The adaptive immune system is one of the body's final defense mechanisms. Unlike the innate response, the adaptive immune system has to be explicitly exposed to a specific pathogen and recognize it as a threat before it can attack. A key difference between the innate and adaptive response is the adaptive has the ability to remember specific pathogens which may invade the body. Adaptive immunity is also systematic, where it is not restricted to a particular infection and can instead fight throughout the whole body at once by deploying humoral immunity and cell-mediated defenses. The humoral immunity dispatches important proteins known as antibodies, which are molecules produced by plasma cells in response to exposure from a specific pathogen and combat viruses or bacteria moving around the interstitial space between cells. The Mycobacterium tuberculosis infection takes place most commonly in the lungs and is transmitted via coughing. When the bacteria in the air is inhaled by a person, they become infected with tuberculosis, which spreads to the lungs. The bacteria is engulfed by macrophages and transported to lymph nodes where they are neutralized. This results in latent tuberculosis, where the person is infected with the bacterium, however doesn't display signs or symptoms of tuberculosis. However, active tuberculosis occurs when the bacteria becomes active and begins to spread around the body. As this takes place, the humoral immunity begins to take effect, with B lymphocytes maturing and developing the ability to recognize and bind to a particular pathogen. Once the bacteria and its specific antibody meet, the B cells are activated in cloning itself and producing an army, all with the exact same membrane-bound antibodies on its surface, designed to fit the Mycobacterium tuberculosis. These B cells become effector B cells, which will fight the bacteria, and memory B cells, which preserve the information needed in case of a future infection with the Mycobacterium tuberculosis. The effector cells mass produce free-floating antibodies required to fight Mycobacterium tuberculosis, which bind to the bacteria and mark them for death by neutralizing the bacteria. This process is known as opsonization, where antibodies tag pathogens for destruction, making it easier for phagocytes to locate them. The antibodies physically block the binding sites on the bacteria to prevent it from hooking up onto the body's tissues. Agglutination occurs when antibodies bind to the bacteria at the same time, forming clumps to prevent it from moving around. The cell-mediated response is a production of T cells which go after body cells that have been affected. T cells cause inflammation, activate macrophages, and initiate other T cells as they regulate much of the immune response. Healthy body cells contain MHC1. Once the Mycobacterium tuberculosis infects these body cells, it becomes fixed to the MHC1 and alerts immune cells of a problem. Helper T cells contain MHC2, which bind to the bacteria of the infected body cell and thus acquires attention of other cells. This process usually takes 6 weeks when a person is infected with Mycobacterium tuberculosis. This graph demonstrates T cell responses are not initiated until 11 to 14 days after the infection of TB. Peaking of T cell response occurs several weeks later, and the bacteria is not eliminated. As a result, this delayed response of the T cells establishes high levels of infection of the Mycobacterium tuberculosis, and thus the bacteria spreads due to no elimination of it, leading to an epidemic. Helper T cells start multiplying, making effector and memory T cells. Helper T cells also release cytokines, notifying the immune system of a problem, with more memory and helper T cells being produced. The infected body cells have bacteria surrounding it, which helps cytotoxic T cells identify it as an abnormal cell, and as a result, the cytotoxic T cells are activated and kill the infected body cell by apoptosis. An additional type of T cell produced includes a regulator or suppressor T cell, which helps prevent B and T cells turning against its own body cells by releasing inhibiting cytokines that tell other immune cells to stand down once the initial threat has been handled, as too many antibodies and cytotoxic T cells could be produced in destroying the body's own tissues. The use of vaccinations for diseases allows for a faster secondary response, as the body has already been introduced to the bacteria. The vaccine, known as the BCG vaccine, is a suspension of living organisms of an attenuated strain of Mycobacterium bovis. The vaccine of this bacteria is effective to a great extent in relation to infants who benefit the most from it. However, its effect on the control of TB has been limited, as this vaccine does not prevent transmission of infection to the individual. In most cases, vaccinations are an effective procedure in preventing the spread of a disease, as they stimulate the production of antibodies for that particular pathogen.